Hello, I am Dr. Wolfgang Winkelmayer. I'm the Chief of Nephrology at Baylor College of Medicine in Houston, Texas. And I have the pleasure to present this trial on behalf of the steering committee. Thank you for the opportunity to summarize the findings from a late-breaking clinical trial that I had the honor to present at the European Renal Association meeting in Vienna, Austria, just a few weeks ago. Titled a phase 2b trial of efficacy and safety of factor 11 inhibition with MK-2060 for preventing arteriovenous graft thrombosis in patients on hemodialysis. And so MK-2060 is a combined factor 11 and factor 11A inhibitor. And the population that was subject to this trial were patients on hemodialysis. So patients with kidney failure, they had to undergo hemodialysis for at least 3 months. And the specific twist here is that they had to have an arteriovenous graft, which is one type of vascular access. Others are central venous catheter or arteriovenous fistulae. They were not allowed in the trial. They had to have an arteriovenous graft. And why is that the case? Because these grafts tend to thrombose. And so it was a phase 2b trial, but it had a hard adjudicated endpoint. And the endpoint was time to the first thrombosis of this arteriovenous graft. But of course, we were not only interested in the primary efficacy endpoint, but also in a broader thromboembolic endpoint that included myocardial infarction, stroke, a new peripheral vascular ischemia, etc. And so it was really also meant to be a preparation for a subsequent phase 3 program in the specific hemodialysis population. The key safety endpoint, of course, was bleeding, and we used the ISTH definition of major or clinically relevant minor bleeding. And the study population, we aspired to enroll over 480 patients. 560 indeed were enrolled, and they were equally randomized to placebo, MK-2060 6 milligrams, or MK-2060 20 milligrams, weekly that is. And the power calculation indicated that the trial duration would be limited to 171 of these arteriovenous thrombotic events accruing. The population that we ended up bringing in, those 506 patients, were very typical and representative of a dialysis population. Average age was around 60 years, equal numbers of men and women. Almost half of the population was Black, which is representative and indicative of the preponderance of kidney failure in the African American population, especially in the United States. And the arteriovenous access was already in place for a very long time in most of these patients, for about 2 years. Primary endpoint, to make the story short, the hazard ratio comparing placebo to MK-2060 20 milligrams was 0.83, and that was not significant. The intermediate endpoint of interest, of course, was suppression of factor 11 activity, and it was very clear that the target was hit hard. The MK-2060 20 milligram group had a median 94% reduction in factor 11 activity from baseline to 24 months in the 20 milligram groups. Of course, the 6 milligram group only had a moderate reduction in factor 11 activity. The primary efficacy endpoint, which was the time to first AV graft thrombotic events, the hazard ratio was 0.87, and that was not significant. The p-value was 0.46. For the other group, for the 6 milligram group, the hazard ratio was actually quite similar, 0.83, of course, also not significant. When we looked at the composite cardiovascular endpoint, which included myocardial infarction, stroke, etc., there was also no significant difference. Now, when looking at the exploratory cardiovascular outcome, the time to first major thrombotic cardiovascular event, The hazard ratio here was 1.13. Again, the confidence limits were wide. They were not expected to be tight, but there was not even a hint or an indication that this endpoint would be reduced by MK-2060 in the 20 or the 6 milligram group. Now looking at the primary safety outcome, time to first ISTH major or clinically relevant non-major bleeding event. Bad news there, the hazard ratio was 1.74, and that was significant. The 95% confidence interval was 1.11 to 2.73. And when we looked at greater detail into the type of bleedings, approximately half of the bleeding events were actually prolonged bleedings from the vascular access, from the arteriovenous graft. And the vast majority, with the exception of one event in each of the three groups, all the other vascular access bleeding events were deemed not serious. And so that's something that, of course, is important. Bleeding after discontinuation of the dialysis treatment session, when the needles are being removed from the axis, it's a problem, you know. Patients would like to go home, they can't because they have to press on their axis. Nurses are eager to get the treatment slot open for the next patient. And so that's certainly, from the patient perspective and from the nursing or system perspective, an important endpoint point. However, if we do a sensitivity analysis and remove those non-serious arteriovenous graft bleeding events, if you will, the excess bleeding gets attenuated by approximately half and is no longer significant. One thing that's important to consider here is that there was a great deal of treatment discontinuation. That's something that is not unusual in trials in the hemodialysis population. Other trials in different fields have shown that. And many of these events are due to mortality. Hemodialysis patients have a high rate of mortality, and so approximately 13% of patients experienced death during the trial. A good amount of patients also received a kidney transplant, but then there were a lot of discontinuations that were owing to physician decision or withdrawal by the patient. And while we don't have certainty from the data, it would be quite plausible to assume that these physician or patient withdrawals were because of prolonged bleeding from the arteriovenous graft cannulation site after discontinuation of dialysis. So in conclusion, in this phase 2b trial of patients with kidney failure requiring hemodialysis via an arteriovenous graft, despite robustly reducing factor 11 activity, MK-2060 did not significantly reduce the time to AV graft thrombosis compared to placebo. And MK-2060 was associated with increased bleeding risk, which was mostly driven by non-serious vascular access bleeds. Further research is certainly needed to understand the utility of factor 11 inhibition generally in patients with end-stage kidney disease receiving hemodialysis.